Today I will talk about the genomic imprinting. I'm originally the behavior ecologist, but I have to struggle with genomes and methylation and a lot of such kind of stuff because the transgenerational epigenetic inheritance is a very important underlying mechanism for the evolution and the caste determination system of termites. And okay. And they shed off their wing and walk on the ground. And they found a pair, and they form a tandem running. And the males follow female, and found a new colony with a monogamous pair. And this is the primary queen, and this is a primary king and mate, and produce first brood. And within 5 years, the queen became physogastric, and the king also developed the testis. And the colony becomes the tens of thousands of colony size. And this is a caste differentiation system of Latigotermes. And after hatching, spend one and the first and the second level instant, they differentiate either within worker pathway, neuter pathway, or sexual pathway. Sexual pathway with other name is nymphal pathway because they already have a wing pad here. And so we can easily distinguish worker and nymph at the third instar stage. And the nymphs develop into the aidates, and they fly from the colony and establish a new colony, independent colony foundation. But if king die or colony die, the nymph pathway, the individuals in the nymph pathway differentiate into nymphoid, the nymph-derived reproductives within the colony. So this is called the secondary queen. But if there was no nymphs within the colony, even workers can have the reproductive potential, totipotency, to become ergatoid. Ergatoid means a worker-derived reproductives. So this is occasionally, in nature, it's very, very rare. But after typhoon or branch, the breakdown, and only workers there, they never give up. The workers still have the potential to become reproductives. It's very rare. And after, as colony development, this guy is a primary king and the primary queen here. And but already we can see the colony size is 1,000 or so after 5 years. And already we can see the neotenic reproductive, secondary, the wing pads here. So nymphoid reproductives, nymphoid queen, supplementary queen here. And after colony development, the primary king and the primary queen still here. But we can see so many neotenic reproductives here. So the primary queen and so many secondary queens mate with this single primary king. And after the maturity of colony development, the king still survive. King have very, very long longevity. But the primary queen was already gone and replaced by the neotenic queens, secondary queens. So please use your imagination, the relationship of this king and these secondary queens. The most of you know, you think about the inbreeding of father, the daughter inbreeding, because colonies started with a monogamous pair, and females never come from outside. So within the colony, these females become queens, secondary queens. So we also imagine that these guys must be the daughter of this king. But if so, most of the colonies is like this. So most of the termite colonies perform inbreeding. How about the inbreeding depression? Inbreeding depression also very strong in termites. So something wrong. So we genotyped the secondary queens and the king. And as you can see here, this king have the AB, right? The two bands here, the genotype AB. But secondary queens never carry A or B allele here. But only single band, but their chromosome number is diploid. So this is a kind of CC or DD, 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 CC, DD, CC, DD, CC, DD. So the the homozygous pair of primary queen is the secondary queen's genotype. So they never carry paternal allele. That means these guys are the parthenogenetic daughters of parthenogenesis of termites produce only females, XY sex determination. So they produce only female. And these females are the parthenogenetic daughters of the primary queen, and not the daughter of the primary king. But they use sex to produce workers and also aids. So they can conditionally use sex and asex. And the secondary queens are all neotenic reproductives produced by parthenogenesis of the primary queen. So the true reproductive system of this termite is like this. They mate with the non-nest mate, and the AB king mate with CD queen and produce sexual eggs. And these sexual offspring, male and female offspring, become workers and aids at the beginning. At the same time, they also perform terminal fusion type parthenogenesis. They produce completely homozygous, almost completely homozygous daughters, CC or DD, not clonal, CC or DD, half clone. And these half clones carrying only maternal chromosomes have a priority to become the secondary queens. So only these parthenogenetic daughters can get this secondary queen position. So CD and CC and DD, the secondary queen and the primary queen, mate with primary king and produce sexual offspring, male and female offspring, and they become workers and elites. At the same time, they also produce asexual reproduction, and asexual daughters can continue the colony. So the queen position is only the succession of queen position is only through asexual reproduction of the primary queen. So actually, primary queen cannot die genetically, and the inbreeding never occur within the colony as long as the primary king survive. So after king die, the secondary king develop within the colony. But after that, the colony soon die because the inbreeding depression is very huge. And so this system is very, very strong top system because they can boost reproduction with increasing the number of reproductives, the queens, without inbreeding, because they can completely avoid inbreeding because queen position is taken over by asexual reproduction. So this is we named this asexual queen succession system, very unique system. But they use asex only within the colony, not the production of elites. So elites needs genetic diversity. They use sex, and the workers also. But only they use asexual reproduction to produce secondary queens. So the first colony foundation is monogamous colony foundation. But at the same time, the queen position is taken over by parthenogenesis, and mating, sexual reproduction produce workers and elites. And after the primary queen die, the parthenogenic queens, secondary queens, produce next generation of parthenogenic queens within the colony, and perform sexual reproduction to produce workers and elites. So genetic diversity never reduce as long as kings survive. Okay, so the selection favors king longevity. So after the evolution of AQS, king longevity, the colony fitness depends on king longevity. King die, colony die. But king, there is king within the colony. So how the termite queens control sexual reproduction and asexual reproduction. At some point, they switch sex to asex because they need to produce a parthenogenetic egg at some point. Otherwise, asexual queen succession is impossible. But how to control sex and asex? We didn't know because these are diploid and termites, so not haplodiploid. So haplodiploid ants and the bees can control the insemination, but termites do not have such a sperm pump or controlling system. But we identified these termite queens control switch sex and asex by using the control regulation of micropile. Micropile is the small opening on the surface of eggshell. And the sperm get into the egg through this small opening on the surface. but the number of micropile varies among eggs. Some eggs have 1, 2, 3, 4, 5, 6 micropiles, 6 openings, 4 openings, 2 openings. But some AQS queen produce 0 micropile eggs. But it's not true in non-AQS type termites, so many micropile. But in AQS type termites, sometimes they produce 0 micropile eggs. So we genotyped the embryo within the 0 micropile egg. And this egg have the 0 micropile. Egg 4 have 1 single micropile, 2 micropile, 3, 4, 5. And we genotyped the embryo within these eggs. And this 0 micropile, the paternal allele cannot get into the egg. So it's only the 202, 202, the diploid, but the homozygous diploid daughter. So these guys are parthenogenetic embryo. So without micropile, they develop as parthenogenetic daughter and take over the queen position. But even with a single micropile, the king's allele can get into the inseminated, and these eggs develop as sexual offspring. Okay. This is a controlling system of the micropile controlling system. And the micropile number is decreased as aging of the queen. So in this colony, the old queen, and we can obtain old queen and the young queen. So these guys are the next generation, the young queens. But old queen, physogastric queens, we get the eggs from these old queens, physogastric queens. The micropile number reduced, and some eggs have no micropile, but the young queens never have 0 micropile egg. So the young stage, they never produce micropileless egg because they just started to reproduction. But as senescence, the number of micropile forming cell reduce, and then at some point, 0 micropile egg occur. And these 0 micropile egg develop as an asexual egg and get new queen position. They become the next generation secondary queen position. Okay, and this type of reproduction, asexual queen succession system, have been identified. The first identified in Japanese term, Reticulatus spiritus, and we also identified the AQS in other subterranean termite species in the US, Reticulatus virginicus, with my friend Edward Bargo, and also my friend identified that type of reproductive system in Italy. And the new type of AQS species continuously identified in the French New Guinea. And the recent review says that asexual AQS system is widespread and higher termites. Reticulatus is lower termite. And completely independently, in higher termites also have a multiple origin of AQS. Okay, so the parthenogenetic reproduction is very, very important reproductive system in termites. And most recently, we also identified completely asexual termite. So completely asexual termite means this termite colony have only female. Okay, so these populations of this termite species have no king. The reproduction is only female. Okay, so parthenogenetic termite, only female parthenogenetic termite was identified in Japan. Okay, anyway, the AQS system is have multiple origin. We look at the Reticulatus, the genus Reticulatus. They originated once in Japan, the Reticulatus spiritus, and another is North America, and one more in West Europe. So 3 times, at least independently, between 10 to 30 million years ago. And also the higher termites, they also have the independent origin. So multiple times the AQS originated. But very importantly, the Reticulatus have the half clonal, so CCDD type, terminal fusion type parthenogenesis. But in higher termites, they have clonal type, central fusion type parthenogenesis. So genotype is different, okay? The Reticulatus produce CC or DD, but clonal type AQS, CD produce CD. But these parthenogenetic daughters have the priority to become the neotenic queens and perform asexual queen succession. Whatever the system, CCDD or CDCD system, the females, parthenogenetic daughters carrying only maternal chromosome can get the next queen position. And also the non-AKS termite also have the parthenogens get the neotenic position much more than sexual offspring. And again, the parthenogenetic reproduction and the caste differentiation. The parthenogenetic reproduction produces asexual nymphoid reproductives, but they also have the ergatoid reproductives, the male and the female, and the nymphoid king. And the nymphoid king and ergatoid king, ergatoid queen and the nymphoid queen, this phenotype of parents can influence on the caste fate of offspring. It's very important. Okay, why parthenogenetic daughters can get secondary queen's position much more than sexual offspring? And this experiment, the other group, they performed this experiment. The father is nymphoid and the mother is nymphoid. The offspring become workers. So the white means workers and the blue means sexual reproduction, sexually developed nymphoid nymphs, male nymph and female nymph. Okay. So nymph pairing produce only workers, but ergatoid and the mother is nymph. So the female offspring become nymph, but female or male offspring become only worker and opposite. And the ergatoid produce 1, 1, 1, 1, 1, 2, 1, 2, 1. Okay, and the nymphoid parthenogenetic daughter become only nymphs and get queen's position, sexual development. Even the ergatoid parthenogenesis produce nymphs and get reproductive position. Okay, so it's a very difficult puzzle. But before, this was misunderstood as a genetic system, but the genetic system cannot explain this because genetic system is impossible for AQS. And but the genomic imprinting explain everything. So the parental phenotype influence on the next generation is not genetic, but transgenerational epigenetic inheritance. And genomic imprinting is very important. Genomic imprinting is the effect of the non-DNA, the factors, epimark, transmitted to parents to offspring through sperm and the egg. And not only DNA information, but also DNA maturation or histomodification or long non-coding RNA also influence on the gene expression of the offspring. And such the effect of transgenerational epigenetic effect result in the for example, the maternal gene, even at the same locus, the maternal gene, DNA sequence is the same. Maternal gene expressed much more than paternal or the opposite. It's due to the epigenetic modification transmitted from the father or mother. And the agent could be the DNA maturation, histomodification, or RNA, non-coding RNA would be the agent. But we focus on the DNA maturation because termite is known already the most highly maturated insects. I skip this. And this is our data of RNA-seq. It shows that these are the epigenetic modification of genes, DNA mature transfer, DNMT3 or DNMT1, and compare the gene expression among the adult or young PK, PQ, or mature PK, PQ, or soldier, male soldier, worker soldier. And we can see the epigenetic marks, establishment of epigenetic modification is completely different among caste. That means the folding of the DNA and the modification of DNA modifications is quite different among castes. And genomic imprinting is originally is the effect of canalizing of the phenotype of the parents. The termite king are super male, and the termite queen is a super female, and developing their testes and ovary much more than the solitary insect. So they establish the epigenetic status to become super male and super female. And the epigenetic modification to realize their adult morphology, sexual status, also the carried over to the next generation to some extent. This is the origin of genomic imprinting, antagonistic action of sexual modification, the factors. after the hatching, the egg develop into nymph pathway or neuter pathway. The neuter pathway, no epigenetic canalization to become reproductive. So sexual development epimarks could be acquired only through the nymphal pathway, not worker pathway. But the worker pathway with one mode, they also become into ergatoid, but they don't have so much sexual development related epimarks. But elates or nymphs have much more epimarks for sexual development. They need to canalize sexual development. And these eggs or sperms from the sexually matured or immatured father or mother or this combination result in the antagonistic asymmetric tug of war action in the offspring. For example, this is the male offspring and this is a female offspring. For example, this male offspring, the mother is ergatoid and the father is nymphoid. So this offspring epimark is pulled toward male side. Development should be pulled toward male side much more than female side, because female is ergatoid, not sexually developed. The male is nymphoid, sexually developed. So the epimark is much more stronger toward the male side. But this one is male offspring, but epimark is modified to become more than female side modification for in terms of sexual development. So this male is pulled toward female side, but they are male, so they cannot go beyond the wall of sex. They're male, but development is pulled toward female side. So sexual development of this male is stopped, and they just develop. That is a worker. So worker is a really neuter caste. And why female, parthenogenetic female can become the queen's position much more than sexual offspring, because they bring epimarks to become queens, double. Okay, they never, there is no the maternal epimarks to become male side. The offspring female daughter carrying only maternal chromosomes, carrying the double epimarks of sex development of female sexual development. So they have the priority to become nurturing queens, much more than sexual offspring. There is no tug of war of epimarks. So antagonistic action of male and female epimarks, paternal and maternal epimarks explain perfectly the empirical data. Okay, that's a very difficult puzzle, but the genomic imprinting can explain that. I have time, enough time. How long do we have?
You have 4 minutes and 4.5 minutes left.
4 minutes.
Yeah.
4 minutes. Okay. Okay. So we are checking the transgenerational epimark. So we perform the different age class primary king and the mature primary king, and king's age also influence on the caste fate of offspring. So that if we made young PK or mature PK, the testis size is completely different, and made with the same the ergatoid queen, and we get the egg and fostering with the other workers, and check the caste status of the offspring. So the young primary king produce much more nymph offspring rather than mature PK. So this is not genotype of epigenetic status. It can result influence on the caste fate of offspring and the female too. So the sexual maturity of king can influence on the caste fate of their offspring. What is the agent? And we compared the metron of the young primary king and old primary king. And we found that they greatly differ in DNA maturation level. And we found the gene region, the mature PK or young PK specific maturation region. We checked the correlation with the caste fate of offspring and the maturation level. And we targeted some gene site of the gene site related to the epigenetic influence on the caste fate of the offspring. And young PK specific maturation genes, and we identified the screening the mature PK specific maturation sites. And so we identified already some genes, the candidate genes involved in genomic imprinting for the determination, caste determination. Okay, I have 1 minute, 2 minutes already?
Sorry, yeah, 2 minutes left, yes.
2 minutes, okay. And but why every termites have such the epigenetic background? Epigenetic inheritance also influence on caste determination, the widespread of termite species. Why? I think this is involved in the origin of eusociality of termites. And but how we know the genomic imprinting involved in the origin of eusociality is very difficult. So with mathematical simulation only the way. And I skip, but yeah, anyway, the subsocial transition from subsocial to eusocial is very difficult enigma, because subsocial stage, everybody finally fly from the colony and reproduce by themselves. But the worker, eusociality is some offspring give up reproduction, but the selection still acting on individual level. How the offspring give up? Some developmental constraint or byproduct of some mechanism before the origin of eusociality generate the lifelong helper. But even with helpers, the reproductive capacity is still low, because the limit of reproductive capacity. So selection favors the helpers nutritionally contribute to the founders, and the selection favors founders produce more eggs because of the additional resource. But selection favors the higher reproductive capacity. Selection favors the higher reproductive capacity to utilize the additional resource. But they need the underlying mechanism, epigenetic mechanism. So more and more they have the reproductive capacity, higher reproductive capacity. They accumulate more epigenetic marks, and the epigenetic marks transmitted to next generation, carrying over effect, can keep the workers at the sexually infertile stage and prolong the worker period. And the prolonging the worker period creates the additional resources. And the additional resources act as a selection force to enhance the reproductive capacity of the parents. And also, so this scheme can generate the origin of eusociality or not. The selection is just acting on the reproductive capacity of the parents to utilize the additional resource. And this, the generation model and the colony model, and we learned the simulation of how this linkage of the parental reproduction and antagonistic action of reproductive related epigenetic marks on the offspring can generate the lifelong helper that is a worker or not. Actually, this model can generate the first eusocial organism in silica, but most importantly, the work performance of the worker is very important. And importantly, inclusive fitness, inclusive selection on inclusive fitness of workers cannot generate eusociality. But selection on the direct fitness of parents can generate eusociality. So the kin selection cannot generate the first origin of eusociality, but selection on parents, parental reproductive capacity result in the origin of eusociality, the lifelong helper. And afterward, the kin selection can accelerate the evolution of more sophisticated eusocial system. But the origin of eusociality is such a byproduct of canalization of epimercs and antagonistic action of such sex-related antagonistic action of epimercs. So my take-home message is now we are shifting from genetics to epigenetics. Epigenetic inheritance, transgenerational epigenetics is very, very important in the next 1 decade or next 10 years. We may identify more and more agents involved in the caste determination, epigenetic caste determination. And also not only identifying the agent, the very important thing is how such the epigenetic system is related to the evolution of reproductive system, and how such underlying mechanism explain the origin of eusociality. The next target is the origin of eusociality. So, okay, time is over.
Brilliant. Thank you so much.
Thank you very much.
Mind blowing. My brain is buzzing. We have some questions. We have very little time, but let's go through the questions. Christina says, are the genes that are differentially methylated in the sperm also differentially methylated in the offspring's genomes, and/or differentially methylated between nymphs and workers?
Mm-hmm. Okay, we didn't check we checked only the sperm, not the paternal gene or maternal gene in the offspring. So we targeted only king, because cytoplasmic factors also involved in the caste determination. But king side, only sperm, nuclei, and also we can exclude the possibility of the histone modification, because histone is replaced by the protamine. So we target the DNA maturation. So we focus on male side rather than female side. And we check sperm, and at least the sperm maturation is completely different between young king and old king. And not only it's very difficult to separate the aging effect and also the sexual maturity effect. It's impossible to separate they develop their testes as aging. So the aging effect and sex maturity effect is very difficult to separate. But at least the aging, different age class kings produce the offspring, which have different type of reproductive propensity. The young kings produce more nymph bias. The old kings produce more worker bias offspring. So this can never explained by genetics, but epigenetics only explain this.
Okay, thank you. A really good answer there. And just one more from Kip, who says, do you think maternal or paternal factors deposited in eggs could explain this phenomenon?
Mm-hmm. Yeah, of course, the maternal effect is also important, but the maternal side is very, very complicated. Okay, the cytoplasmic factors, many kinds of cytoplasmic factors, but the king side is, important thing is the king effect is really significant. So king side is simple compared to female side. So our first target is king side. And we know already the sperm maturation influence on the, correlated with the caste determination of the offspring. We know already. So the next is the female side. So we know the sperm aging of king influence on the sperm maturation. That influence on the caste determination. The next is the egg side. The egg side is more complicated, but we should do that. Next.
Okay, thank you, Kenji. I'm really sorry. We're going to have to move the rest of the questions into the chat on Slack.
Yeah, thank you very much. Yeah.
Really awesome talk. Thank you.